Okay, so yeah, pathogen biology. So I'm going to give you some definitions of terms and jargon and so forth. I'm going to describe the multifactorial nature of bacterial virulence, and I'm just going to use a little rhetorical trick. Every slide is going to have a title starting with S, so you can remember the steps in successful infection and describe the various molecules. And as we pointed out the other day, these kind of issues do come up. You might say, well, this is a bit arcane trying to define what is virulence. It's a philosophical issue. Yeah, but your supervisor, your examiner may well decide to pick on this in the first 10 minutes of your viva. And if you can't, if you just say, I don't know, I never thought about it, I don't care, it will colour the way the viva turns out, perhaps. Why is that OneDrive doing that? Just go away. Oh my God. Just stop it. Right, so bacteria colonise body surfaces. So in topological terms, they live on the outside of us, even though you might say the inside of your gut is the inside of you. Topologically, it's not. And they can be commensal or neutral. They can constitute the normal microbiota or the normal microbiome. They can cause infection in where they penetrate host defences, enter tissues and multiply. And as we mentioned before, they can cause clinically obvious disease, or it can be subclinical infection. Pathogens is the name given to microbes that cause infection. Some people use this term pathobionts. I think it's quite an ugly term and I don't ever use it, but the point is that some pathogens can colonise you without causing disease. And some potential pathogens that cause disease in one body compartment can live in another body compartment without causing any trouble. The commonest example of this is organisms that live in the gut, but don't cause disease in the gut, but do cause disease when they spill into another body compartment, such as the urinary tract or into the bloodstream. So an E. coli that's capable of causing a urinary tract infection in the gut behaves just like a commensal, just like any other E. coli. It's only when it gets into the urinary tract that it becomes a problem. And opportunistic pathogens are ones where they won't generally cause disease in normal people. There has to be some way in which your defences are down. And that can be that your immune system is actually compromised. But in fact, our primary line of defence against infection is intact physiology and anatomy. So if you've got third-degree burns covering a large part of your body, you are effectively immunocompromised, and opportunistic pathogens will infect you. And the severity of disease depends on this property known as virulence of the pathogen. The more virulence the pathogen, the smaller the infectious dose needed to establish infection and cause disease. So if we take an organism like a encapsulated Streptococcus pneumoniae, you can kill mice if that's the sort of thing you like to do. With just 100 cells injected into a mouse will kill it. Other organisms, you might have to give 1 million cells to actually kill, because it's just a matter of statistics. And there are very few pathogens where a single cell of that particular pathogen introduced into the body is able to cause disease. There's always a kind of numbers game here. Pathogenesis is the term that we describe the process leading to infection. And virulence factor, well, this is a difficult one, but a feature or structure that contributes to the ability to cause disease. But we'll see that that's quite a slippery term, really. And then we've got various portals of entry, so the various mucous membranes, gut, respiratory tract, genitourinary tract, the skin, and some pathogens can enter parenterally if there are ruptures in your bloodstream that allow organisms to get deep into your tissues, then they can do that. Now, if we're going to talk about virulence factor, we have to sort of recognise this is a rather slippery concept, and I mentioned this briefly in the previous talk. Is this something that's needed to damage tissues? So is it kind of analogous to a weapon, if you like, an offensive weapon? or is it something that's just needed to colonize tissues? And I mentioned Stan Falco's molecular Cox postulates, often now called Falco's postulates, where he says a specific gene should be consistently associated with the virulence phenotype. When it's inactivated, the bacterium becomes avirulent. If the wild type gene is reintroduced, the bacterium should regain virulence. If you can't do genetic manipulation, and there are still a few organisms where we don't, I mean most organisms we can, then if you can show that antibodies, for example, against that particular factor actually neutralize pathogenicity, that can be a proxy for it. So here's a picture of Stan Falco. There's the last time I ever encountered him, where he joined us virtually for the opening of a new centre, bioinformatics centre in Warwick. In fact, you were there, weren't you, for that?
I was there for that.
Yeah, exactly, yeah. And it was great to see Stan joining in. He was at that stage too unwell to travel, and anyway, it was only for a short meeting. And he died a couple of years after that, unfortunately. So, you know, this is a fuzzy hype concept. It's one of those things, if you want to get your paper, you know, jazz it up a bit to get more attention, get a press release, you know, you want to say, oh, it's a virulence factor. And I say, yeah, what's a weapon? What counts as a weapon? And the issue is, you know, is why do you want to know? So, interesting kind of factoid is that the United States government treats strong cryptography as a weapon. And there was a time a few years ago when people were told that if they took out this particular encryption algorithm, the RSA encryption, out of the United States, they were exporting a weapon and they were breaking the law. And so a lot of geeky people actually put a version of that algorithm onto a T-shirt and wandered through customs with it and so forth, just to be provocative. And it's an interesting question, you know. So if cryptography is a weapon, you know, is the ribosome a virulence factor for an organism to cause a bacterium to cause infection? It has to have a functioning ribosome. If you took out the ribosome, it wouldn't be able to cause infection anymore. Does that make the ribosome a virulence factor? And these are kind of reductio ad absurdum arguments, but they do sort of point out there's no really hard answers to this question. And it depends why you want to know. So why do we want to know? If you want to develop a vaccine or a drug, you want to target things that might be considered virulence factors. Similarly, if you want to devise a diagnostic approach, you want to target something that will separate out the virulent forms of that organism from the non-virulent forms. So, for example, Corynebacterium diphtheriae, you want to know that it's carrying the diphtheria toxin, the gene for the diphtheria, and making the diphtheria toxin. And so it's quite legitimate to say, oh, well, I'm going to call that a virulence factor and target it in my diagnostic assay. And then there's this evolutionary perspective, and we mentioned this the other day as well. So if we take the simplistic view, you know, we say infection is like a war, and we can actually take some bacterial exotoxin. So we can take components that are made by the bacterium, and we can purify them away from the bacterium, and we can inject them into an animal model, and they will reproduce the signs and symptoms of the infection in that animal model. And there are many examples there: tetanus toxin, botulinum toxin, diphtheria toxin, anthrax toxin. And this view that, okay, so we've got the virulence factor in our hand, we can now do something with it, has borne fruit dramatically in the early 20th century. Because what people did was, well, we've got the virulence factor, and if we just inactivate that so it doesn't cause disease, but it still looks the same to the immune system and produces an immune response, we've got a vaccine. This was highly productive in terms of eradicating diseases like diphtheria from the population, and tetanus and so forth. And all of us have had toxoid-based vaccines as part of our normal vaccination schedule. But, you know, this is perhaps a simplistic idea that, you know, all you need to cause disease is a toxin. You know, all you need to cause trouble is a grenade. just lob the grenade and off you go. And this just shows you, just to illustrate the point, this is what happened to diphtheria in England and Wales once we introduced vaccination with the toxoid vaccine. And you can see that there was absolutely astonishing drop from this being a serious cause of serious disease and actually a cause of death in this country, to one where now, I haven't looked at the latest figures, but it is pretty much always an imported infection, and we'd expect to see half a dozen or a dozen cases a year from travellers returning from overseas. As I say, I haven't looked at the literature lately. I'm not aware. If there are homegrown outbreaks, they usually only involve a handful of people. There have been outbreaks in living memory, like 30, 40 years ago, but this is, the toxoid vaccination has got rid of that. A more sophisticated view is to say, well, actually, virulence is a multifactorial process. And this graphic here is quite a famous graphic that illustrates the Napoleon's army's march on Moscow during the Napoleonic Wars, and what actually happened to that army in terms of the number of people who got to Moscow and the number of people who got home again. And it greatly illustrates the fact it wasn't just the armaments that they took with them to Moscow. It was their ability to keep the army supplied with food and with water, to keep the army safe from infection, safe from the cold. All of these things played a decisive role here, rather than the weaponry that they had. And Napoleon's supposed to have said, and I haven't checked whether he actually said this, an army marches on its stomach. And so this more sophisticated view, which has come in the last couple of decades, 2 or 3, 4 decades, sees this as a multi-dimensional approach as well, where virulence is actually a program organised in time and space. So if you think about E. coli has to live in your mouth as you're swallowing it. It goes into your stomach. It has to survive the acid bath in your stomach. When it gets out of the stomach and into the small intestine, suddenly all these bile salts are thrown at it. And then eventually it might get through the small intestine into the large intestine. And at each of those stages, it's going to be facing different challenges. And so there are multiple steps in successful infection, and I'm going to just take you through those now. I've been rather fanciful in making each step begin with S, but it might help you remember them afterwards. So the first thing is, the old saying, sex comes before disease. So bacterial sex is what drives the evolution of virulence. And we've already mentioned the idea that by having sequences, we can draw a tree of life that includes bacteria. This has had practical consequences, and we can identify and characterize non-culturable bacteria and place them on that tree of life and develop diagnostic tools. And what we saw with the advent of bacterial genome sequencing was that there's, in some parts of that tree of life, there's been rampant horizontal gene transfer. And some people have actually gone so far, Didier Raoult, the man I mentioned the other day, is one of them, as to say, there's no such tree of life anymore, no such thing. It's all nonsense. We shouldn't be saying tree of life. It's a web or a network of life. It's not a tree. Darwin's thinking is stupid, Some people say that. There are plenty of other people who still say, no, we can use this tree-like thinking to devise databases and taxonomies and so forth. But yeah, there is this idea of genomes as mosaics. There's a core genome and an accessory genome. And those cores are the housekeeping genes, the things you need just to be a bacterium. You need to make your ribosome. You need to make your cell envelope. But the optional stuff, the accessory genome, may be niche specific. So this particular form of E. coli needs that if it wants to live in a river, or it needs that if it wants to live in the bloodstream, and so on. And there all these. This is just undergraduate stuff. But just to recap, there are 3 different ways in which they can exchange DNA. Transformation, they can just take up the DNA from the environment. Transduction, where phages will carry DNA into the cell. And there is this weirdness that many phages do carry things that we would call virulence factors into the cell and help the bacterium. And then there's conjugation, which allows them to acquire things like plasmids. There are also transposons. I mentioned the other day when we were talking about repetitive DNA in bacterial genomes. So these jumping genes, the simplest form of those are so-called insertion elements or insertion sequences, where they just carry a gene that allows them to jump around. But there are more complex elements that are transposable, can jump around, and in addition to carrying the transposase that allows them to jump out, actually carry virulence genes. So, for example, the enterotoxin genes that you find in E. coli are actually carried on transposons. There are virulence plasmids that carry whole suites of genes into pathogens to give them a complex phenotype acquired in a single genetic leap, if you like. And then I've mentioned already the phage-encoded virulence as well. And this is just an example from my own research paper, one of my highly cited papers from 2006, where we went through trying to find the virulence factors that were targeted by a particular secretion system in the chromosome of this so-called enterohemorrhagic E. coli, OMA57. And what we found when we did the bioinformatics analysis, and this was done by Nabil's, the guy who did it went on to go supervise Nabil's PhD shortly after he did this when he went back to Australia, is that you can see that these are the prophages, these bacteriophages that have integrated into the chromosome. And if you colour them by GC content, you can see that there is an abrupt change where they go from generally being AT-rich to GC-rich in the genes within them. And those ones coloured green are these particular virulence factors that are carried at one end of these prophage genomes. So it's a kind of interesting phenomenon that you see these things scattered throughout the genome. I mentioned already the idea of pathogenicity islands, and this was a concept that initiated when people started studying uropathogenic E. coli. They said, oh, there are these bits of the chromosome which can be deleted easily, and they seem to carry more than one virulence factor in them. And this, when people started doing comparative genomics, they realized this was actually quite a common theme, that you'll get a block of genes in one genome that's been acquired en bloc as a block that are not present in another genome. And where those differ between pathogenic strains and non-pathogenic strains and actually confer that virulence phenotype, we call them pathogenicity islands. They're a specific example of a more general class that we might call genomic islands, where you look at the difference between 2 different genomes, you'll see a whole bunch of genes have been shoved in, or sometimes deleted as well. So these things tend to carry virulence genes, more than one usually. They're present in pathogenic versus non-pathogenic. They often have this different GC content from the host chromosome. So every bacterial species has its own typical GC content. Are you familiar with the idea of GC content? Yeah, so it's a signature for that genome. It has its own characteristic GC content. And things that have just recently arrived in that genome tend to have a different signature, have a different GC content. This is one of those things where people argue about, is this adaptive? Is this just the fact that they've come from a different context? But you do see these differences. They're quite, I don't know if we're going to look at them later on Artemis. Are we going to look at GC content? I'm not sure. We can. And you can see it's actually quite striking often that you'll see a block and the GC content just varies. These are typically dozens or hundreds of genes, of kilobases. They're compact units, often flanked by direct repeats and tRNAs and ISs. Often might be a bit strong. They are sometimes. I think often it's quite cryptic as to how on earth did they arrive in the genome, how did they get in there? and do you see mobility genes or not? And the early characteristic that sort of marked them out, that they could easily be deleted, it's not actually quite so common when you look across all of these pathogens to the islands. So here's an example. So this is a locus called the locus for enterocyte effacement, or LE, which is a pathogen to the island found in enteropathogenic and enterohemorrhagic E. coli. It encodes a type 3 secretion system, which is a kind of molecular syringe that the bacteria use to inject proteins into eukaryotic cells. And you can see on this graphic here two important features here. So you can see when we colour the GC content that it drops quite dramatically as you go through this island, and the genes flanking it have a different GC content. You can also see that when you align the chromosome of E. coli 0157 with the common lab strain K12, you can see that there's just this cassette that's just been shoved in there. On either side, all the genes that are there in K12 are also there in 0157 in the same order, same length, very similar sequences, but then you've got this discontinuity where this thing has been wedged in. Okay, so that's enough about sex and pathogens in the islands. Bacteria need to sense the environment, so they sense all sorts of things going on: nutrient availability, temperature, osmolarity, cell density. And in simple cases, there may be just a very simple thing that they're detecting. So what we know is that the diphtheria toxin gene is regulated by sensors that sense a fall in intracellular iron levels, and this actually relieves repression of the toxin gene so that the toxin gene becomes activated. In more sophisticated cases, we have these very complex signal transduction cascades, and there is sensor mechanisms, there are response regulators that actually, where there are cascades, and in a sense, this is where we start to see the pathogen. It's more like a cruise missile, if you like. It's actually sensing its environment. It's knowing where it is. It's responding to the environment in appropriate ways to encourage its survival as it goes through. And we're sensing and switching virulence factors on and off. And so in bacteria, almost all gene expression is regulated. There are some things you might say, well, you need that all the time, but those are relatively rare components. Most genes are inducible. There are a few that are constituted on all the time. And basically this is, it's a bit like, why do you leave your lights on in the house? You turn them off if you're not in the room. It's a waste of energy and resources for the bacterium to be making things it doesn't need in that particular environment. And you might say, well, yeah, but it's marginal, isn't it? But one of the things that Darwin pointed out, grain in the balance is what settles. If cells are competing with each other in a particular environment, even small gains in their ability to control use of resources, it can be useful to them. So you get coordinate gene regulation. So you have operons, as we've mentioned before. There are these jargon terms, the stimulon and the regulon. So stimulon, you get a bit, sometimes mention that. So, people spend a lot of time studying how does a bacterium respond to oxidative stress. What happens if you take a bacterium and dunk it in hydrogen peroxide? More commonly, you'll see the term regulon, which is basically a whole bunch of genes that are regulated by the same master regulator. So just like in your house, you have a switch to turn the lights on and off in a room, but there is also a fuse box where you can turn off, I'll turn off the whole lighting circuit, I'll turn off the whole heating circuit. And this is the way in which bacteria regulate their genes as well. They do that. And so they coordinate their regulation of virulence in response to the signals that they're encountering within the host. And this is often a multi-layered hierarchy that they have available to them. They can have just changes in the DNA sequence, so they can, the dose of a gene can go up. There are genetic rearrangements, so we can see a regulator that's turning a gene on can flip over, so it gets switched off, or it actually flips over from expressing one gene to regulating expression of another gene. So Nabil showed me a blog post he did the other day about serotyping with O and H antigens. The flagella systems in some organisms like Salmonella, they actually have what they call two phases, where you can flip the gene, the regulatory components of the gene, so that they will turn on one flagella gene or another flagella gene. There's transcriptional regulation, where you have activators and repressors. Many of these have this helix-turn-helix motif. They bind to DNA and they influence the transcription of that DNA. There are changes in folding and stability. You can get translational regulation, so whether the protein is actually made, or actually post-translationally, whether that protein remains stable or whether it gets degraded quickly. All of these things make a difference. And then finally, there is the potential to covalently modify proteins, and this is part of many of these signal transduction pathways, where you get phosphorylation in a very common motif you see is these two-component sensor regulator systems. How much of this have you come across in your undergraduate teaching? Are you generally familiar with it? Yeah, okay. So I'm just reminding you. We won't spend too long. Here's one example, the toxar regulon in Vibrio cholerae, where there are various environmental signals. You've got this toxar regulon that is regulating multiple genes and proteins and systems associated with virulence, which includes the toxin, the cholera toxin, but includes a lot of other stuff as well in terms of adhesins and such like. SWIM, many bacteria are motile, pathogens are motile. Proving that motility is crucial for virulence has been tricky, but there are cases where it is. So upper urinary tract infections, where the bacteria get up into your kidney and cause pyelonephritis and can spill into the bloodstream, it's been shown that they need to be able to swim up from the bladder to cause that. And the usual organelle of motility is the flagellum, but there are other variants as well. And the flagellum itself is a very complex organelle, a very interesting case study in the evolution of complexity. Bacteria need to stick. So one of the primary ways in which we get rid of bacteria from places we don't want them is peristalsis. And basically anything in your gastrointestinal tract is getting swept through and goes out the other end. And so the bacteria, if they want to stick around, they have to stick to mucosal surfaces. If they want to survive in your mouth, they have to stick to mucosal or to teeth in that situation. And there are various ways in which they can do that. They have to be able to scavenge nutrients. So one of the other things that we humans do to try and prevent bacteria ever getting a foothold in our tissues is that we keep iron levels extremely low in our body fluids. And if you get ill, if you get COVID or you get some other kind of infection, and this induces what we call the acute phase response, where you get a bit of a fever and so forth, that is accompanied by a drop in iron in your body fluids as well. And we know that if you have iron overload, and there are some people with a genetic predisposition to accumulate iron, those people are more susceptible to certain forms of infection. And so bacteria are exquisitely sensitive to this. They use things called siderophores to actually grab hold, chelate in the jargon, they collate the available iron and transport it into the bacteria. And they have ways of ripping the iron out of iron-binding proteins from the host directly. And this is coordinately regulated as well. So in E. coli, one of the key regulons is the so-called fur regulon. Some bacteria go one step further, and they get around this issue by making sure that they don't need iron. So iron is, a common cofactor in enzymes. Treponema pallidum, which causes syphilis, has managed to create an entire repertoire of enzymes that don't use iron, uses alternatives, and so it gets around it that way. And we know that iron is also crucial to some of these aggressive, virulence factors. I've mentioned diphtheria toxin, also required to regulate sugar toxin production, and Pseudomonas aeruginosa exotoxin A. A key theme in research in pathogenesis is survival of stress. And in fact, Mark Webber gave a talk yesterday about stress survival in biofilms. You know, it's still a very hot topic. So there's all these other stresses that, you know, if you think about it, what you see when you look at bacteria in the lab is a very weird artificial state. Because to get them to grow as colonies on agar, you make life as lovely as possible for them. You give them the lovely temperature that they want, you give them loads of nutrients, you give them just the right atmosphere, make everything great. But in real life, when they're in the body, they are under stress. There's acid stress in the stomach. It's interesting. It's again, one of the key defenses we have against gastrointestinal infection is all that acid in your stomach. You take that acid away, you will become more susceptible to GI infections. Fever is a defense. So some bacteria do not like being forced to grow at a temperature slightly higher than body temperature. Within phagocytes, you know, they pour reactive oxygen radicals onto bacteria as a way of destroying them. And one of the things, very early on actually, 40 years ago, when people were saying, oh, what are the major pathogen antigens? So if you look at something like TB, what we want to know is what antibodies do humans make against TB? And they wanted to know for two reasons. One is they just wanted to understand the process of infection better, but they wanted to see if they could make a blood test for TB by just looking for antigens. And what they found was the predominant antigens that they were seeing antibodies against in the human bloodstream were actually stress response proteins. They were heat shock proteins. They've been defined as heat shock proteins, but they were in fact generic stress response. They were so-called chaperonins. And this was a bit of a surprise, but looking back, it's not that surprising because basically all bacteria are under stress when they're in the human body, particularly when they're causing disease. And we know that detoxification proteins play a role in virulence. So when I was doing my PhD, I discovered a superoxide dismutase from Salmonella carried on a phage. It's one of those proteins that's actually involved in making the organism more virulent. And as I said, when you have no acid in your stomach, you're more susceptible. And that happens in some people as they get old anyway, but there is this huge kind of experiment in our society at the moment in that a large percentage of the population is taking acid suppressive medication, taking proton pump inhibitors, which actually changes their susceptibility to infection. It changes also their gut microbiota, but it does change their susceptibility to infection. Stealth, you have to hide away from host defenses. So some bacteria produce what we call IgA proteases, which are specific proteases that target the key antibody that's found on mucosal surfaces, IgA. They also bind to immunoglobulin so that they effectively are grabbing hold of the immunoglobulin that's supposed to be grabbing hold of him and them and holding it the wrong way round so it can't actually do anything. Many pathogens have these characteristics that they can resist complement and opsonization. Many capsular organisms, pneumococcus, Klebsiella pneumonia, Haemophilus influenzae, Bacillus anthracis, pestis, all of that. And the bacterial surface is often coated with proteins that will actually, or polysaccharide that will actually help with this process. And Mycobacterium tuberculosis, coming back to that, it has a cell wall that actually prevents it from being degraded after phagocytosis. Another thing that bacteria do, not just bacteria, but viral pathogens and parasites do this, is they vary their antigenic profiles as a way of confusing or evading the immune system. This is a very common tactic. And in bacteria, this involves things like LPS, capsules, pili, flagella. And this is why things like serotyping probably work, in that the bacteria are evolving variation in their surface repertoire to avoid the immune system. That is one answer. The other answer is that there may be other factors. There may be avoiding predation by phages or amoebae or whatever, but basically that does help. And there are a variety of mechanisms for that, we won't go into that. They can adopt a cryptic niche and hide away. They can engage in antigenic mimicry, so group B meningococcus has a capsule that looks very much like sialic acid that coats many human cells. And this huge antigenic diversity also helps with that. So here's an example from in my career where we sequenced Campylobacter jejuni as part of the team that did that, and my friend Brendan Wren followed this up by showing what you get in many places in the Campylobacter jejuni genome is you get these so-called homopolymers. So you get 8 Gs in a row or 9 Gs in a row. When you try and sequence that, and you try and do what you did the other day at an assembly, and then you look carefully at all the reads, you realize that you cannot get an unequivocal consensus across these. And what you find is, for example, a third or a half of the reads will have 8 Gs, and a third or a half will have 9. If it's a third, some of them might have 10. And what is happening actually within that population growing on the plate, there is phase variation because there's slippage. As the DNA polymerase replicates these regions, it sometimes skips over one or adds one in when it wasn't there. And you can actually colony blot these. So you can see that these are, so it's actually come out of alignment, but basically you can see that there are some colonies there that light up with cholera toxin. When cholera toxin is applied, it's actually binding to some of the surface moiety here, and you can see that there are some colonies that are nice and black, and there are some colonies that are just greyed out because it's not binding. Strike back. Okay, so what about actual aggressive virulence? So endotoxin is a key component in many bacteria, and then there are the exotoxins. So there's this key difference between endotoxin and exotoxin. The exotoxins are soluble secreted proteins, where endotoxin is a component of the bacterial cell wall. And endotoxin is a key component of gram-negatives, and it is able to trigger, particularly the lipid A part, this toxic part, is able to trigger a number of effects. Pyrogenicity affects the white cell, so white cell rates go down and then they can come up again. If you get large amounts of endotoxin in your bloodstream, your blood pressure drops, and we call this endotoxic shock. It's actually a feature of gram-negative shock. So in the early days when people tried to give intravenous fluids, stick a cannula in your arm and say, give you some saline, it was bedeviled by the fact that it was very difficult to get rid of endotoxin that was just contaminating the products. And you'd run in some saline, and the patients, instead of doing them good, you'd actually make things worse. And this is a key point when we're dealing with septicemia with gram-negatives, that the patients go into this kind of shock. Exotoxins are secreted proteins, typically with enzymatic activity. They have various effects. They can kill cells, they can interfere with nerve function, they can interfere with the function of the gastrointestinal tract. And as we mentioned, antitoxin antibodies provide immunity. There are toxoids that you can use as vaccines. Some of these are exoenzymes that actually just chew up things. And some of these can be really quite nasty. So here's gas gangrene. And within gas gangrene, one of the key components of these phospholipases that just chew up the membranes of the host cells. Coagulase is an enzyme produced by Staphylococcus aureus that produces clots that kind of wall off infection to save it. And there are various others as well. Some toxins actually punch holes in cells, form pores. So there's those so-called hemolysins. There are the zinc metalloendoproteases, and you came across these the other day. So I just threw you an example of a potential one of these that had just been isolated from cow shit and nobody knew what it meant. But the ones that have been well characterized, botulinum toxin and tetanus toxin, they're zinc metalloendoproteases that go into nerve cells, and they have slightly different effects on when they cleave synaptobrevin to actually cause disease. There are some toxins that get put inside cells, and they are actually, if you like, there's a kind of payload, which is the toxic part, but a large component of the toxin is actually a delivery mechanism to get them in. So there's a translocation of binding subunit, B subunit, and active A subunit, and this is the case with diphtheria toxin. And interestingly, just, sort of understanding these kind of toxins, here are AB5 toxins where there are actually 5 binding subunits and 1 active subunit. The understanding these has actually given people a new way in, so that what people are doing is that they can separate out the binding components and the enzymatic components, and they can swap them around to try and, say, target cancer cells with toxins that only go into the cancer cells as a way of killing them. So it's an interesting example where having a full and detailed understanding of how disease is caused gives you tools. I mean, obviously the biggest kind of payoff from this side effect spin-off is botulinum toxin, which is used in a billion-dollar industry for cosmetic reasons. But understanding how that toxin worked was a necessary precursor to that. Then there are these so-called pyrogenic exotoxins, superantigens that you get in, say, Staph aureus. I'm nearly finished now. How are we doing for time? Yeah. We've got these secrete and subvert systems, where secretion systems, these are complex systems that made of multiple proteins that translocate bacterial proteins, or in just a few cases, they sometimes translocate DNA from the protein into host cells. And we've learned recently that some of these not only involved in warfare between bacterial cells and eukaryotic cells, but things like type 6 secretion are actually a mechanism whereby bacteria joust with each other and inhibit each other as well. So it's very interesting. And one of Stan Falco's famous phrases was that bacteria are excellent cell biologists. They've actually developed all these systems for subverting the normal function of the eukaryotic cell. And they target all sorts of processes, the cytoskeleton, inflammatory responses, tight junction barrier function, cell cycle, mitochondria. Each of these, you can find kind of virulence effectors that are targeting those processes. And some people, the guy who was a postdoc in the lab when I was a PhD student, Gary Frankel, he's effectively become a eukaryotic cell biologist, and he's had to learn all the tools. Cynthia Whitchurch, who runs our microscopy suite, is also in effect a kind of cell biologist now, because you have to learn all the tricks of how to label things, do the microscopy and so forth, that people simply studying eukaryotic cells on their own have to do as well. Bacteria have to survive within cells, and the intracellular lifestyle is quite a common approach among bacteria. And there are a variety of mechanisms that allow bacteria to survive within host cells. And then they have to be able to spread through cells and organs. So, if you're a typhoid bacillus, you want to get around in the bloodstream, you can live inside macrophages and be transported around that way. If you're traveling in the blood but naked, you have to be able to resist complement. Some bacteria will survive within cells, and move between them. So Shigella and Listeria monocytogenes both manipulate the actin cytoskeleton to move around. And then finally,
You have to pop next to me.
once you've caused disease, you need to get out of that host and into the next host. And so how do you get out? And normally they depart through a specific route. And the portal of exit determines dissemination. So obviously it can be respiratory, it can be on the skin, can be in feces, urinary tract, bloodborne infection. And there's a whole raft of kind of philosophical issues here as to why do bacteria cause disease in the first place? And there's this idea of balanced pathogenicity, and this has come to the fore also when people are discussing SARS and SARS-CoV-2 and COVID. Is the natural state of things for there to be a balanced pathogenicity where it's not really in the interest of the pathogen to cause any more trouble than it needs to just to get on, and that being too virulent is a bad thing for the pathogen. Some people would say that high virulence is a sign of recent emergence of a pathogen, and that pathogens have to evolve towards symbiosis. That's what some people say. Other people say, if you ever follow Alan McNally on Twitter, he'll give you a whole bloody load of verbiage about there is no certainty whatsoever that subsequent variants of SARS-CoV-2 will be less virulent or evolve towards being less virulent, and it all depends. I think I've got that in the next slide. There are counter arguments to this. So, if you depend on causing a huge, if you need to be spread by a biting arthropod, a mosquito needs to come take some blood from the patient and take the next patient. It's in your interest to be as plentiful as you can be in the blood so that you get picked up by that mosquito. So if you end up damaging your host, that's kind of, put that to one side. It's better to and so severe disease and death in the host. Death, obviously, you can argue, well, that's an end point for the bacterium or the parasite, because once the host is dead, it's lost its home. But if it deftly moves on to the next host, it doesn't care whether it's causing bystander damage as it goes through. If you're cholera, Vibrio cholerae, and you want to infect the next person, actually producing the most prolific diarrhea that you can so that you can spray yourself out into the environment, well, that's in your interest, surely. And the fact that occasionally some of those hosts that you've infected die, well, it doesn't matter because it's a hit and run thing. You've run on to the next patient. We know that there are some pathogens that just don't travel from human to human, but they cause disease in humans. So how is it they're tooled up to cause disease in humans? Legionella. Why does Legionella, when it gets in your lung, cause such a nasty pneumonia when its normal life is living in water? What's going on? And we mentioned that earlier in the previous talk about this eco-evolutionary view, that it's tooled up like that because it's used to encountering unicellular eukaryotes in its environment that often will predate on it, like amoebae, and it has all that stuff as a way of coping with amoebae. But there's another, we are getting a bit philosophical here, but there's another curious point, which is that you may know that Richard Dawkins' second big book was called The Blind Watchmaker. It's the idea that evolution doesn't have a goal in mind that's far away. All that natural selection is doing is equipping that population to survive in its current environment. It doesn't know what's coming next. So if a person has meningococcus in their throat, it's living there as a commensal fine, we know some populations of meningococcus will actually get into the bloodstream, will get into the nervous system, will proliferate, and actually kill the host. You get fatal septicemia or fatal meningitis. And you try and think, well, how is that benefiting the bacterium? It's going into the skull. The skull is a closed vessel. It can't get out the skull once it's in there. It's not helping it to be transmitted further downstream. And the argument that people put forward is that at every stage it's optimizing its success of surviving in that environment. So the few cells that spill into the bloodstream will suddenly evolve to be within the blood to survive as best they can and proliferate in the bloodstream. And they're not interested. They have no way of knowing that this is going to damage their ability to get to the next host. They just keep going until they overwhelm the environment. Another interesting point, and I've gone on a bit too long, but vaccines. We mentioned diphtheria toxin vaccine. The toxoid vaccine is what happens when you give the toxoid vaccine to a population? What you'd expect to see is that Corynebacterium and diphtheriae that's producing the toxin, they would no longer have any selective advantage, but you would still have everyone carrying the non-toxigenic versions. But what we actually saw was the Corynebacterium diphtheriae as a whole went down in the population. And there are interesting arguments about that as well, but I think we'll move on. Anyway, there we are. I've just taken you through a quick whistle-stop tour of how a pathogen is able to cause disease, and that might give you some conceptual background for when we start to look for so-called virulence factors in genomes, that you might do that with this kind of conceptual background in your head. And you should be entirely skeptical of those people who say, oh yes, I found 153 virulence factors in this organism, Well, it's not that precise a science, and it all depends on what you decide.
Yeah, there are some fairly asinine publications that just say, we did a metagenome and the number of antimicrobial genes or the number of virulence factors are increasing over time, as you go down the river or whatever. And you sort of think, well, that's interesting, but doesn't tell you very much. And we didn't, Mark sort of is talking bigger topics, but majority of what he's saying also applies to antimicrobial resistance.
Yeah.
So who's actually has that in their project, antimicrobial resistance as a key topic? Anyone? Surprised?
No, we haven't got anyone doing that. We've got people working on development of drugs. And again, that's all of that stuff, as you will know, it makes sense only in terms of ecology and evolution, because the antimicrobial resistance genes that are giving us so much grief these days, they actually evolved in the organisms that make the antibiotics in the first place. So the antibiotics are a way of bacteria seeing off the eukaryotes around them. The antibiotics were there to see off the bacteria which they are competing with, but obviously they don't want to get intoxicated by their own antibiotics, so they have to make their own antidotes to stop themselves getting hit by the antibiotics. And it's those kind of antidotes that have become the antimicrobial resistance genes that we see that have evolved. So, and again, there's a whole raft of literature on this. It's got like a game of rock, paper, scissors. If you're a bacterium and you don't produce an antibiotic, and you don't, then you're going to be at a disadvantage in a population where some of them are making antibiotic. So you can make an antimicrobial resistance gene that protects you from the antibiotic, and you can make an antibiotic. But what is best? Is it best just to have the antimicrobial resistance gene so that you're not affected by the antibiotic, but you're not wasting any time and any energy making the antibiotic? Anyway, it's an interesting topic. There's some further reading for anyone who's at all interested in any of this further, and that's basically that finished. And I will stop recording as well. Does anyone have any comments or questions?